Good morning and welcome to today's technical press briefing. This is not one of our usual press briefings that we kind of held to discuss public health emergencies and EMA's response, but a technical briefing that is focusing today on RNA technology. So due to the pandemic and the mRNA vaccines against COVID-19, many people are meanwhile aware somehow of this technology, as the vaccines have shown the efficacy and versatility of RNA technology against viral infections. But there are many other areas where it also holds great promise, such as cancer and rare disease. During today's briefing, we will provide you with information on possible applications of the RNA technology and scientific and regulatory challenges, and what EMA can do to support the development of RNA-based medicines. The briefing follows a regulatory and scientific virtual conference on RNA-based medicines that EMA has organized yesterday. During this conference, regulators, academics, and industry representatives have shared their perspectives on the potential and the challenges of RNA technology. And today you have the opportunity to ask any questions on RNA you might have. I'm Marie-Agnes Heine, the head of the Communication Department of the European Medicines Agency, and I'm delighted to introduce to you our two speakers today, who will be able to offer very different perspectives on this exciting technology. Here with me today are Professor Annemieke Aartsma-Roos from the Leiden University Medical Center. Annemieke is Professor of Translational Genetics at the Department of Human Genetics in Leiden University. She is one of the leading researchers in developing RNA therapies for patients with rare genetic disorders. She will be able to give you deep insights into the science and is uniquely placed to talk about the academic perspective. Annemieke is joined by Professor Stefan Tierstrup, our Chief Medical Officer at EMA. He chaired the workshop yesterday and can offer the regulatory perspective on many of the promises and challenges of RNA technology. Initially, our experts will provide you with their perspective on research and regulation of RNA technology, and then we will open the floor for questions. We expect that this briefing will last approximately 45 minutes. As usual, your microphone is disabled for the duration of the briefing and will be opened for colleagues who have their hands raised in WebEx. The briefing today is not broadcast live, but we will make the recording available afterwards. I'm now giving the floor to EMA's Chief Medical Officer, Stefan Tierstrup. Stefan, please.
Thank you, Marie-Agnes, and good morning and welcome. Thanks for joining this press briefing. As already said yesterday, we had a conference organized by the Agency on RNA Technologies. The conference was well attended by approximately 800 participants from a broad range of stakeholders, and that is pharma industry, academia, patient, and other regulatory authorities. The aim of the conference was to stimulate further development of RNA technologies in the interest of patients and public health. We wanted to identify challenges and opportunities for developers in academia and in small and big pharma, and to understand how we as a regulatory authority best possible can engage with them and support them in their development. Before turning to the content of the conference, it might be good for me just to briefly explain what RNA is, and we can take further clarification questions on these technologies in the Q&A session. So RNA is short for ribonucleic acid and is present in all living cells, not only human cells. Their key function in cells is to act as a messenger carrying the instructions from the DNA in the genes to produce proteins. So they are the code for proteins. As medicines, and already alluded to by Marie-Agnes, we all become familiar with the messenger RNA vaccines, the mRNA vaccines against COVID-19. In this case, the mRNA in the vaccines are carrying the instructions to produce the spike protein, which sits on the SARS-CoV-2 virus and against which our immune system reacts. So the technology can obviously be broadened out to carry information for production of other kinds of proteins in our body, either to stimulate the immune response or to replace proteins that are not produced by our cells, maybe due to a disease. However, RNA technologies and antisense oligonucleotides, which we hide under the same terminology, goes far, far beyond mRNA as we know them for the vaccines, and can be used as an example to silence the production of specific proteins, so diseased proteins that are caused by genetic diseases. Since the mRNA vaccines for COVID-19 has been so intensively discussed in the past couple of years, we decided to exclude any discussions on mRNA and RNA technologies against infectious diseases from yesterday's conference. We also deliberately excluded any discussions on a specific technology where small strands of RNA is used for gene editing, such as in the CRISPR-Cas19 technology. Back to the conference. The conference identified a number of promising applications of RNA technologies. I won't go into detail, but just highlight diseases of the brain and spinal cord in a broad range of diseases, including genetic diseases. Cancer immunotherapy was another important point discussed. We discussed both more off-shelf cancer immunotherapy, but also very personalized cancer therapy where individual patient cancers were used to develop therapy. These therapies are sometimes referred to as cancer vaccines, and I think it is important at this stage to be clear on this terminology. We already have vaccines against infectious diseases such as the human papillomavirus, the HPV vaccine, authorized. These vaccines prevent the disease, but also prevent the cancer that may develop as a consequence of having had that infection. When the terminology is used in the context of RNA technology, cancer vaccines are not referring to vaccines that can prevent cancer, but actually products that can stimulate the immune system to improve the fighting of an already existing cancer. So I prefer using the term immunotherapy for cancers because the term cancer vaccines can be misunderstood. Other challenges discussed in yesterday's conference were manufacturing, not only scaling up on manufacturing capacities when you go from research grade into commercializing your product, but also the challenges that goes with manufacturing products if you're only treating a very few patients, which is the case for many of these very rare diseases that you can target with this technology. We also had an intense discussion in many of the sessions on so-called platform technologies, dealing with platform technologies, and we can go into details what a platform technology is in the Q&A session, but platform technologies for manufacturing, but also very important platform technologies to either fully or partly replace the use of animals for testing of new medicines. We also discussed financing and in particular pre-competitive research done by academia and how to obtain financing of this. I'll stop here, just concluding that yesterday's conference, at least from my perspective, were very successful. We identified, not surprisingly, that there is a need for continuous dialogue between developer and regulatory authorities and us as the European Medicines Agency. We have mutual interest in developing these products for the benefit of public health. I will pass on to Annemieke Asma-Rusch from Leiden, as already said, and renowned and internationally recognized researcher in the RNA technologies, and Annemieke was part of yesterday's panel. Please, Annemieke.
Okay, thank you, Stefan. So before going into my highlights of yesterday's meeting, a brief introduction of the work I've been involved in the past, over 22 years. So as Stefan explained, the mRNA, the RNA contains the genetic code for protein that the cells in our bodies make. And the technology I've been working on is called antisense oligonucleotides, or ASO. And that actually targets the mRNA that's made by the body itself. And you can do that targeting for two things. You can silence an mRNA so that the protein it codes is not produced. Now, why would you want to do that? Sometimes due to a genetic mutation, patients will make a toxic protein, and that toxic protein then causes eventually cells to die, which is of course a bad thing. You can imagine if that happens in brain cells, that's going to lead to functional loss. Now, if you target these toxic codes with antisense oligonucleotides or ASOs, you can reduce the amount of toxic protein that's produced, and obviously that will then lead to therapeutic effects. Another thing you can use them for, and that's what I've been involved in mostly, is if mutations make the code unreadable, the protein cannot be produced. And you can use the ASO technology to make the code readable again, and then patients can produce a missing protein or a partially functional missing protein instead of having no protein at all. So this technology is actually quite advanced, and it preceded the mRNA vaccines. And currently there's already several of these ASOs approved by the EMA. So going back to the meeting, I think having the meeting was good. And as Stefan already outlined, it was a multi-stakeholder meeting. So there were representatives from industry, from the regulators, from academia, and in the audience there were also patient representatives. And I think it's really good to have this dialogue, because if we have a dialogue, then we can learn to understand each other. And I think what was clear, as Stefan already hinted at, this platform approval or platform technology, how to streamline things more. I think so, since I work mainly on a rare disease, so the antisense oligonucleotides primarily target rare diseases that are caused by having a mutation in a single gene leading to a toxic protein or protein missing. There, streamlining things is really important because you have very small groups of patients. So you can't do the massive clinical trials. You have a very limited group of patients. So that means that if you can build on what's known from other diseases using the same technology, things can be speed up and streamlined. So building on this knowledge is really important. And that's something that was discussed, and that's something that well, the regulators, industry, and academia all see as being important. Another thing that was discussed is learning from failures. So it's important not just to share successes and things that work, but also to learn from things that do not work as hoped, or that turned out not to be effective, or that turned out not to be safe. And I think as an academic, we have a role there to publish about this so that everyone can learn. And then one final thing that was discussed. So for industry, it's very common to go to the regulators for regulatory advice. For academia, it happens a lot less. And I think I'm, well, an example of an academic researcher who did start that interaction with the regulators and who did start that dialogue. And I can say from personal experience, our first interaction, I'm an expert on Duchenne muscular dystrophy, and we and some other experts, we went there and we thought, we're going to tell the regulators how they need to think. And that's not how it works. So after having this dialogue, we saw, okay, it's a bilateral education. We can tell them about the peculiarities of the rare diseases that we're working on. Regulators can't be an expert on all the rare diseases, but they have to tell us how the regulatory system works, because only then can we anticipate as academics. And even because often the compounds that companies will develop, that industry will develop, often it starts out in academia. And if we can proactively already see, well, what do we need if we start to develop these compounds? By having the interaction with the regulators, things will be streamlined as well. So I think I would also make a point of stressing that academics definitely should start having their interactions with regulators early. And from me, from personal experience, I've done this a couple of times, and it has benefited my work a lot.
Thank you very much for these introductions, both of you. And we are now going to open up the floor for questions. So, dear colleagues from the media, please turn on your camera if you want to ask a question. And as usual, please click on the icon at the bottom of WebEx to raise your hand. And when I give you the floor, we will unmute you and you can ask your question. And let's see the questions. We have received one question from Euronews Serbia. I know, Tara, that you are on the line, but I can also read it out here. So it's a question from Euronews Serbia asking, some studies have shown that personalized mRNA vaccines have been more effective in treating cancer. Will this approach to cancer treatment come faster, or a generalized mRNA vaccine against a specific type of cancer? Please tell us more about how personalized mRNA vaccines work. As we said before, this briefing is mainly about RNA technology for medicines, but I'm sure that my colleague Stefan can tell you a little bit about this topic. Over to you.
Thank you, Marie-Agnes, and many thanks for the question. I already in my intro alluded to the term cancer vaccine and how it works. And there are basically, at the moment, two types of cancer vaccines. It has taken a long time to figure out how to target cancer with this technology, because cancers are developing within the body, and therefore cancers hide from the immune system by having a lot of surface proteins in common with the patient's body. So the ability to find targets that are unique for the cancer and not for the rest of the patient's body, so to speak, has been a challenge. So these cancer tumor-specific antigens is where the research is going, and they are increasingly being able to detect. So the technology now goes into two things: either tumor or cancer antigens that are located on every cancer, so to speak, at least every cancer within a type of cancer, let's say within lung cancer, just to pick an example, that are common among all patients. And you could develop an RNA technology where you stimulate the patient's immune response to further react against that antigen and therefore kill or help killing the cancer cells. So that will be a kind of off-the-shelf RNA vaccine. Sorry for using that word, I'm not fond of it, RNA therapy against the cancer. The other development, and that's where the real big promise is for the rare cancers, is that you can actually take a biopsy of the cancer in an individual patient, and in the laboratory then do genome sequencing where you can identify protein structures that are unique for that cancer in that particular patient, and then quickly develop an RNA therapy against that individual patient's cancer. So actually using the individual patient's cancer type to develop a highly personalized therapy. So the development goes in those two directions at the moment. I can't say what comes first here. Of course, there's I think there's a commercial interest in both. It may be on a broader scale interesting to develop these therapies to be an off-the-shelf, because the very individualized one will be most likely very expensive and will not be something that you can offer in each and every hospital. So there'll be some constraints in which kind of patient who will get access. It's something that has been studied for the mRNAs for the past 1 or 2 decades, and was actually the reason why industry was so quick to develop mRNA vaccines against COVID-19, because they had already a lot of knowledge about this platform, so they could easily switch from having tumor antigens or having mRNA coding for tumor antigens to have the spike protein mRNA in the vaccines. So that was actually the reason why industry was so quick to respond to the COVID-19, that a lot of cancer research has been going on. It has not really met the goal yet. There are more research to do. You might have seen in the press that some companies are teaming up with the UK research system and UK Cancer, and they have a nationwide genomic database that may help them identify these targets. So it's very promising. Difficult for me to say when we will see the first product licensed.
Thank you very much, Stefan. I do not see any question at this moment, but perhaps I can ask one because I think it would be interesting for the public is how many RNA medicines are already approved in Europe? And perhaps can you give an example in, I don't know, perhaps first Annemieke?
I think so. There's different types of RNA medicines. I think the number differs on whether you include the mRNA vaccines, because then it's 10, or whether if you just say, well, the antisense oligonucleotides are the ones that target the RNA that the body itself makes, then the number would be 8. And I think probably the best example is Spinraza or nusinersen. That is an ASO for spinal muscular atrophy, and that's a very severe disease. So, well, there's different severities, but the most severe type causes patients to die within 2 years or to be on permanent ventilation. And that's because they miss a specific protein, and the treatment with the antisense oligonucleotide can restore the production of that protein. And depending on how often or how quickly it's given, so either before the symptoms or after the symptoms, if it's given before symptoms arise, then the therapeutic effects are really quite big. And patients normally don't learn to sit, they never start walking. And after treatment, there have been patients who learned how to sit, who started to stand, who started to walk, and who, well, the patients who've been treated longest now are treated for a couple of years, where normally we know they would have died within 2 years. So I think that is a really good example of the potential of the antisense oligonucleotide or the ASO therapies, where in this case really it had a really good therapeutic impact. And that one was approved in 2017 by the EMA, and I think was one of the first antisense oligonucleotides to be approved.
And I may add to that that answer that we have had roughly a little less than 100 interactions with industry and academia on these kind of products, which to me sends the signal that this is an area of huge interest. So we are not completely starting from scratch here, but there's definitely more to come. And as I alluded to in my introduction, there are a lot of potential therapeutic applications for diseases where there currently is no treatment, and actually many of them very severe diseases, including diseases of the brain and spinal cord, just as the example was just given here. So very promising.
Thank you very much to both of you. We now have a question from Ian Schofield. Ian?
Okay, good. Yeah, you mentioned that the conference discussed both the potential and the challenges of mRNA technology. Would you be able to give a quick overview of the key regulatory challenges you've discussed in terms of mRNA medicines?
Stefan, please.
Well, there are many challenges. First of all, if we take it from the way we think as regulators, the manufacturing, how to produce these products, and how to generate the sufficient evidence to document that you can manufacture these products in a consistent manner. A very lengthy discussion around this. Also, the so-called platforms, and maybe it's time to discuss this a little bit. Annemieke alluded to this. The fundamental concept about having a platform technology is that this is a platform. This is something we know. It's something we've done before, and from a regulatory perspective, this is something we have been assessing before. So we know what the challenges and the pitfalls are, where potentially where we need to be more vigilant. So companies using platforms, they can streamline their development process because we as regulators are much more comfortable with that part of their dossier, and we can focus on identifying issues, challenges, problems in the part of the dossier that is new, where they add something to the platform. So, but again, platforms are many different things, and I think that's also a regulatory challenge to agree on platforms, to have platforms, to agree on the platform and to get them approved or accepted as a common understanding. And that feeds into something that is outside our world, because there's also some interest in making platforms as a priority right for industry, so you will actually have a patent on some of these platforms, which will be a challenge for academic researchers. Finally, I think we are also, I think one of the outcomes and some of the challenges is that we are moving, to Annemiek's point, into a field where we as regulators may not be on top of the science, and therefore we need to engage with those who are on top of the science to understand that and come forward with the regulatory proposals to solve some of the issues. And finally, and maybe that's on the edge of your question, I think yesterday we had a lengthy discussion about, is there a need for us as regulators to develop more guidelines and put guidelines on a page, which on one hand is a yes, we need more guidance. The problem in developing guidance in a fast evolving scientific field is that when the guidance come out, it's potentially outdated because it takes time to write the guidance, to agree on it, have it in consultation and so forth. So one of the take-home messages yesterday was perhaps we should be better at doing something a little bit quicker, some kind of Q&A documents that can be more living. And to Annemiek's point, we are very interested in talking to companies and academia, not necessarily within the framework of a formal scientific advice, but also in our innovative task force where the discussion is a little bit more free-flowing.
Thank you very much. Annemieke, you want to compliment?
Yeah, maybe I can add a little bit. So, of course, I'm from the academic perspective, so not really the regulatory challenges per se, but still, so coming from the rare disease field, and we're now even trying to develop ASOs for personalized cases, so individuals, there's regulatory challenges there as well, because if you go back again to the manufacturing challenges, so there's a challenge if you want to scale, but there's also a challenge if you want to do this for just a single patient, because, for instance, you would need 10 grams to treat a patient lifelong. And then how do you manufacture in such a way that it's high quality without having to make 20 kilos to do all the quality checks, etc. So those are things that we face as challenges. And I think, again, as Stefan said, having a dialogue with the regulators about these things is very good, because on the one hand, so it's an approach with a lot of potential. There's approved compounds now. There's many more in the pipeline in clinical trials and in preclinical research. So more are coming, but still it's a relatively new approach. So we know that probably each ASO is more similar than it is different, but we need to also have more data to feel more comfortable about when we can say, well, okay, this is a platform and we know about this. So I think we need to start having this dialogue now so that we can anticipate these things rather than, again, looking back and then deciding it is a platform when actually it's already there for a while. So I think having this ongoing dialogue is important. And I think especially from the academic perspective of now trying to develop these things for individual cases, it's been really productive having interactions with the regulators, because of course they have a lot of knowledge because they confidentially see all the information from the individual companies, but that does give them the helicopter view that they're not telling us like company X has done this or that, but they can give general perspectives that only they know because they have this helicopter view.
Thank you very much. So I do not see any hand from the colleagues from the media up, but another question that came up, and you already alluded to, Stefan, is, the importance for RNA to reduce perhaps the need for animal testing. So how can this technology reduce that need, Stefan?
It's actually inherent to the diseases that you're going to treat, because many of these diseases are so rare and so intimately linked to the genetics of humans that you cannot create an animal model. In classical pharmacology, you have a lot of animal models where you test your compounds in early stages in these animal models. So animals having a disease or carrying a disease that is similar to humans, or somehow maybe the animal has been modified to have a disease similar. This is not the case for these conditions that you target, many of them because they are genetic diseases. So by that, it's already inherent that you have a challenge that you cannot do this in animals. So you need to find other ways. And there are a lot of new technologies here. Some of them are called organ on a chip, where you simply use human cells which are grown in the lab, and you can then do the testing on human tissue in a kind of synthetic organ. It's not a living organ we're talking about, but we're talking about cells of humans, so you can do the toxicology testing there. So a lot of new technology, which of course will be developed and are being developed for these technologies, but would on a longer scale have an impact on how we develop medicines in every area, and ultimately could reduce and maybe in some cases completely abandon use of animals.
Thank you very much. As there's still no question, I take the opportunity to ask another one myself, which is about perhaps about safety, because this was a topic that was really very big when the mRNA vaccines against COVID-19 were developed. So how safe are the RNA technologies? Is it possible to say something general about it?
Maybe I can start. I think so. There's different ASO tools, so to say. So you can restore a missing protein, you can reduce the toxic protein, you can provide the mRNA. So each of these tools have their own characteristics. So that's one thing that you can say, well, we know for each of the tools, we have a rough idea of what the safety is, but it also depends on the tissue that you want to target. Because, for instance, the nusinersen Spinraza spinal muscular atrophy ASO that I mentioned, that one is injected intrathecally, so into the spinal cord fluid. That means that it will primarily go to the central nervous system, and the exposure to the rest of the body is very limited. Of course, that gives a different safety profile than if you would inject something into the bloodstream, where it will go mainly to the liver, for instance, but also to other tissues. So it's not so much as general safety, it's really on a case-by-case basis. But then if you say, okay, so we know now this is the safety profile if you inject this type of ASO into the spinal cord fluid, then there you can say, okay, so we can build on that data when we develop additional ones that work in the same way. And the same for, well, this is one that we inject into the bloodstream that will then go to the liver. Again, if you have then additional ones, you can work and you can base on what you know already about the safety profiles. So I think you can't put them all in one basket, but definitely because we have these different types and we know roughly about the safety of each, we can build on that, and then we can anticipate the safety profiles.
Thank you very much, Stefan.
Just to add, I think we can take the mRNA vaccines for COVID-19 as an example. I, of course, acknowledge that there are adverse events with these. We can take the mRNA vaccines. I acknowledge that there are adverse events, also severe adverse events, but it's important to remember that these are extremely rare. So fundamentally, this technology seems, with the knowledge we have today, to be generally safe. There will, of course, be safety issues linked to depending where you administer your product. Annemieke mentioned products that are given into the spinal fluid. Of course, that carries a risk in itself to inject something in there. There's a low risk of infections related to the procedure. But more, I think what the picture we have today, that the adverse events is mainly linked to the site of administration. And a lot of the research today also goes into making these products organ-specific, organ-targeted, so you make sure that the therapy ends up where it belongs, so to speak, and not everywhere in the body. These are challenging products to work with because RNA is in itself, if you just use the RNA ourselves, that our cells produce themselves, have a half-life it disappears from the blood like, dew for the sun within very, very few seconds. So you need to put these medicines into capsules. You need to modify in a way that it can find its way into the tissue where the therapy is supposed to work. And that, of course, to some extent limits the the possibility to cause side effects outside the target organ, and therefore the adverse events are more targeted to the organ or related to the organ where the therapy is targeted.
And maybe a final comment to that. So most drugs have side effects, and not just the RNA therapies, also other drugs have side effects, but you should also see this in light of the risk of having the disease. So again, I'm from the rare disease field, where often patients have very severe, progressive, debilitating disease. And then, while knowing the risk of not treating, if the risk of side effects, if the risk of not treating is much, much worse, is death within 2 years or losing all bodily functions within 5 years, then of course you're willing much more to take on the risks of the side effects. So I think that is really important to bear in mind, that even though these things will have some side effects, like all drugs, if the risk of non-treating is so severe, then generally patients are much more willing to also take the risk of side effects or uncertainty of side effects.
Thank you very much. So as I do not see any additional questions, I'm going to close today's technical press briefing. So thank you very much for your interest, and thank you very much to our experts today. And as always, if you have any further questions, please don't hesitate to contact the press office at press@ema.europa.eu. So thank you very much and goodbye.